let's start again. Here's a Blackboard site. Here's video lectures. I'm not a really great TV producer, and hopefully it's gonna get better by the end of the semester, but if video lectures are a little bit of help, I would be glad if you can just take a look and use PowerPoints or video lectures. Right there. Your grades will be also uploaded on Blackboard, so you can, as soon as I grade it, so use this site. Video lecture, and here's kind of our last one, me looking a little bit scared. Here's my channel. So some small tutorials are here, but I plan to do a little bit more. Okay. So now shut it down. Please pass your quiz in. Got stuck. Okay, give it to me. All right. Thank you. All the questions after the lecture. I have enormous time to spend with you after the lecture. Or if you have some questions, email me so we can set up the time to talk. The subject of today's lecture is DNA, is chapter 21. Why all of a sudden we're starting with DNA today? Because it's the fourth macromolecule, and it's better to learn it now because all molecular genetics, all molecular biology, all medicine is based on DNA research now. And if you learn it now, hopefully the whole human biology will become a little bit easier for you to learn, because there is a bunch of concepts that are used in human biology that are based on DNA. Okay. What are the four macromolecules of life that we studied last chapter? Carbs. What else?
Lipids.
Lipids, or another name for lipids is fats. Proteins and DNA. So, which one are energy? Carbs and fats. Very good. And proteins, what is the function of the proteins in the human body? Enzymes, metabolic activities, personality. And that's why DNA codes for proteins. You have lecture notes, and here's your vocabulary. And for a long time we were going with a small lecture notes, 2 pages. And then I realized that there's a bunch of concepts that need to be kept in mind. So I created another piece of paper, concepts that are based on DNA. They're all very important. So, first concept, and could you please open lecture notes? Right here. One camera. So, because we're going to go concept by concept. First of all, what you have to keep in mind is that DNA is a macromolecule. It means it's huge. It's huge, it means that it consists of subunits. What are those subunits? Something connected to something, connected to something, connected to something, and it's very, very, very long. So, the subunit of DNA is called nucleotide. Now, nucleus, because DNA is kept in the nucleus. It's, well, it's actually DNA is nucleic acid. It's not because it's kept in the nucleus. There are 4 different subunits. You know, in case of carbohydrates, there's only one type, glucose and glucose and glucose. In case of proteins, there is about 20 different amino acid, amino acid, amino acid. In case of DNA, there is 4 different types of subunits. So, now question to you. Who watched the movie Gattaca? Yeah! Cool movie. All the subunits are here. 1, G, A, T, C. And behind this letter, there is actually a name for subunit. Guanine, adenine, thymine, cytosine. But if you just remember the letters, the abbreviation for those subunits, that is already great. So what else do we see here? Okay. What is a subunit? Nucleotide. How many different kinds of DNA subunits exist? Got it? 4 different types. Now draw a nucleotide. Each nucleotide has 3 parts. Take your coloring pencils so you can see those 3 different parts nucleotide more easily. And the most important part, a coding part of each nucleotide is called a base. It's not the base. Biology is very confusing. It's not the base that acid or base thing and so on. The base is like in the baseball, something that is really important. This is a coding part, and this base can be G or A or T or C. Then there are 2 other parts. And we draw a pentagon here. It's similar to glucose, but it's not glucose. It's chemically, it's another type of carbohydrate. Just draw a pentagon. And in your book, it's called sugar. It's not sweet. It's not our table sugar, but it's chemically it's called sugar. And then a phosphate. Phosphorus. This part, phosphorus and sugar, is similar in all of the nucleotides. Let me show you the picture. If you have your book with you, this is a picture on page 484. And what it shows, see, base, sugar, phosphorus, base, sugar, phosphorus, and so on. And they all are connected to each other. So phosphorus and sugar are the same for all of the nucleotides and are used for connection. And base, you see letters here, bases, the base part of each nucleotide is a coding part. Okay, this was the first concept. Now, as you can see, DNA is shaped as a double helix. Double helix means two spirals, just like that, okay? That's how it should pretend that you are a DNA. Double helix. Two snakes that are wrapped around each other. How are they held together? With a hydrogen bonding. This hydrogen bonding that exists between two strands of DNA. They keep it together, and then those strands could be easily pulled apart. And then if you let it go, they snap back. This mechanism is very useful for getting information from DNA when you need to make an extra proteins, if you need like bigger muscles or something. You need to separate the strands and then they just snap back. So what is drawn here are hydrogen bonds that can exist only between complementary nucleotides. And in this case, A is always complementary to T, and G, we say it's complementary to C. Between A and T, you see here, there are two hydrogen bonds. It's just chemical structures that can form those hydrogen bonds. And between G and C, there are always three hydrogen bonds. And that's what holds DNA together. Okay, now we'll look at this picture. And this is an important picture. So when you work at home with the material, look at this picture. You may not need to read everything around it, but do look at the picture because our brain learns faster when it deals with the picture rather than with text. Okay, so we got through nucleotide. Question?
Can we, on concept 1, where it says which parts are the same between the nucleotides, can you go over that again?
Sugar and phosphate.
Sugar and phosphate.
Sugar and phosphate.
Is phosphate, which part is different, or is that the same?
Sugar and phosphate are absolutely the same between all of nucleotides. This is A or T or G or C.
Okay.
And they're similar because they need just to connect them together. They are not a coding part of the nucleotide. Only those bases that you see in Gattaca, they are coded.
Which part is different?
Coding
Base.
part, base.
Okay, thank you.
Base. Thank you for your questions. Okay, now next concept. Okay, how the nucleotides are connected to each other with the help of phosphorus and sugar parts. Each phosphorus of one nucleotide is connected to sugar. That is chemically possible, so we will get this long chain. Now, concept 2, DNA is a double helix. Two spirals held together by hydrogen bonding between A and T, and G and C nucleotides. What is a hydrogen bond? This is a bond between molecules. In comparison with ionic bond and covalent bond, hydrogen bond is a bond between different molecules. Now, in order to reproduce, you need to create a copy of DNA for your offspring, right? Because when you're passing your genes to the next generation, where DNA comes from. So this structure of DNA allows very precise replication. Replication is a scientific word for DNA duplication or doubling of the DNA. If you look at your vocabulary, in another kind of lecture notes, you will see the word replication here. So please put there copying of DNA into DNA or doubling, precise doubling of the DNA. Because you don't want to get your genes, your genetic material changed or damaged when it is being passed to a new generation. You also see the word double helix. This helix, double helix means two spirals, two molecules of DNA connected with each other with a hydrogen bond. Two spirals of DNA. You see the word genome. Like you probably heard human genome, cat's genome. This is a sum of all genes of the population, if we are talking about human population. So this is the sum of all genes or genetic variations that we can see in humans, or in cats, or in bacteria. Now let's look at the mechanism by which our DNA could be doubled precisely. And this is a picture of it's right here. Page 486. So what happens here is a double helix. And you see that C always, we say, matches complementary to G. You should expect to see G in the other strand of DNA, or T always matches A. So if one strand carries A, the other strand will carry T, because the hydrogen bond could be only formed between A and T, and G and C. So then, to double a DNA, we have to pull it apart. What you see here, there's special enzyme, special protein that pulls it apart and builds it by using one strand of DNA as a template. So let's try that. You are an enzyme. And first thing you have to do, take double helix, pull it apart. Now you look at one strand and you see like C here. What are you going to put against C? G. Next one, A, T. And the enzymes do it very, very quickly. They don't need to think a lot. Then the first enzyme puts so-called complementary nucleotide, and then another complementary nucleotide, what you see here on both strands. Then connect those nucleotides together and just keep going forward. There are another bunch of enzymes that come and check the precision of the job, and that's how DNA is doubled. So what I'm trying to say is the very structure of DNA allows precise doubling of DNA and passing your genetic material to future generations in non-damaged grade form. So see, we already are on concept 3, DNA replication is building the exact copy of DNA. What do you need to replicate DNA? You need one strand of DNA, you need an enzyme, and you need a bunch of free nucleotides, right? So just 3 things. What is the product of DNA replication? Second strand of DNA, or doubling DNA. And that's, by the way, how you grow, how you develop, and how you heal your wounds, because if you get a cut or something, you need a brand new cells. The brand new cells have to have the same genetic material as the old cells. So the neighboring cells just keep dividing, and in order to divide, they need to double their DNA. So if you were an enzyme, how do you replicate DNA fast and without too much thinking? You just put complementary nucleotide against the strand using this rule. Please write down this rule. This is very important. Now, concept 4. Your DNA is the code for making your own proteins. Your carbs and fats are not coded by DNA. DNA codes only for the proteins. The question is how 4 nucleotides can code for so many complicated proteins. Okay, anybody knows how to work with Morse code? Beep, 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 beep. Okay, the emergency, like SOS, you remember. 3 long beeps, 3 short beeps, 3 long beeps, 3 short beeps, right? It's by 3. DNA goes like Morse code 3. Nucleotides will code for 1 amino acid. This is another rule I would like you to remember. 3 nucleotides code for 1 amino acid in the protein. This is absolutely the coolest thing about molecular genetics that makes everything very simple. 3 nucleotides in DNA code for 1 amino acid in the protein, and that's why we call it triplet code. So, see, what happens here? You take 1 strand of DNA, and what it means to read a genome or to read a gene is to find out a sequence of nucleotides in 1 strand of DNA. So I'm putting like, why don't I write the word Gattaca? And it goes for a long length. We forget about sugar and phosphate. In this case, what matters are those coding parts. And one of the easiest concepts, one of the very simplified concepts that I would like you to remember, that one gene codes for 1 protein. Of course, there are some non-coding parts of DNA which are used for regulations. But let's say we get a very simple gene and it codes for one protein. How do we know the sequence of amino acids in the protein? By reading DNA code. We just find the start of a gene, and then we mark every 3 nucleotides. And this is a specific code that had been deciphered. And let's say in this particular case, the combination ATC codes for amino acid number 7. They all have names, those amino acids like leucine and tryptophan and something like that, threonine, alanine. But let's say this combination will code for one amino acid. By the way, I would like you to get your coloring pencils and do something like that. Different colors. And then you put the word code or anything, so that helps you to remember that every combination of nucleotides will code for some specific amino acid in the protein. Here is like table. I just would like you to take a quick look at that. You don't need to study it or something. But this is the ciphered code of DNA. You see the names of amino acids here and the DNA and combinations of nucleotides and the DNA that corresponds to a particular amino acid there. Okay. Any questions? Is everything clear so far? Is everything clear? Okay, let's go. So, as you can see, the sequence of amino acids in the protein is predetermined. And a bunch of genetic diseases are related to the fact that if something happens to your DNA, the sequence of amino acids, oh, by the way, draw the circles, little circles here, mark it as a protein, and you can see that DNA not only codes for specific amino acids in a protein, it also codes an order of amino acids in the protein. So what is a genetic disease? Something happens here, let's say during the replication, C here became an A, enzyme made a mistake. Now this amino acid, let's say, is no longer amino acid number 7 or tryptophan, it's something else. What happens with the shape of protein? Cannot work. This is important. That's what makes the structure of protein will make it an enzyme. It's like, you know, you have 2 legs and 2 arms, and all of a sudden you have only 1 leg because this amino acid got changed, got broken. You can't walk real fast. The protein can't work. It cannot do the job of removing some debris from the cell or running metabolic activities as it should. And it's being passed to the next generation. That's what genetic disease is, and that's why all molecular diagnostics is based on reading DNA code, while the disease itself, while the symptoms itself are related to non-functioning, malfunctioning protein. Okay? Because if you know the sequence of DNA, you can understand you can understand why the protein is malfunctioning, what needs to be changed by genetic engineering or concept. Okay. So this was concept 4. Your DNA is a code for making your own proteins. Why do we need to make proteins and not carbs and fats? Why? Why we don't code for carbs and fats? Why do we code for proteins?
Makes everything.
Yes, because the proteins are like, I want you to think about the cell and about the proteins. Proteins are little people of the kingdom. Everything depends on them. Every kind of job in the human body depends on proteins. We have the same carbs and fats. It's just, lumber or building blocks or bricks, or we don't need to code for them. We just need to eat them. As a table sugar, as a honey, as another fat. We eat them. We build proteins. Okay? From the amino acids that we eat with the steaks and so on. So carbs and fats are the same among all of you. Proteins are different. That's why we call it proteins. Now, what is the material for making proteins? Amino acids. You build the protein from amino acids. How do we get this material? We eat steak. We eat someone else's protein. Well, some amino acids could be synthesized in the body, but we also like to reuse some other, beef and pork and some other proteins, disassemble those proteins into amino acids, grab those amino acids, build our own proteins. Because you eat a steak day after day on a regular basis, you're not looking like a cow, you don't grow arms. You use those amino acids to build your own hair color, eye color, your own personality. Yeah, and if you eat fat and carbs, you basically get the same results. Proteins are amino acids linked, I'm just reading this, linked together in the correct order. How does the cell know the order? The order of your own proteins is coded in your genetic material. See? Because the cell is going to make a protein according to your genes. Okay, what exactly DNA codes for? DNA codes for specific amino acids in the protein and the order of these amino acids in the protein. Why do we call DNA code a triplet code? Because 3 nucleotides in DNA will define one amino acid in the protein. And that's how the code works. Now, we talk about gene expression. It's like gene can be silent, it can work or not work. Sometimes the gene is there, but the regulation doesn't work, and the person still doesn't have a protein, and so on. So the word gene expression, which is on your vocabulary, or genetic expression, is all the steps from DNA code to building protein. And this is molecular genetics, molecular biology in a nutshell that we're gonna cover in 20 minutes. Let's open the picture. Actually, I spent most of my life studying this particular thing, how to move genes between different organisms, how to make them work, how to make a functional gene, and so on. Because I came to US as a biotechnologist to work for University of Virginia, and we studied Drosophila. It's a little fruit fly, but because our genome kind of 66% similar, so you study the genes in Drosophila because it multiplies every 2 weeks, so you can see the result of what you're doing to your gene, and then you can make extrapolations or some conclusions related to humans. Like for example, the gene that I studied was affecting, you're gonna laugh, the position of little hair on Drosophila wing. But similar gene in the human will affect how your limbs grow. So if the humans have the same gene not working, those babies die within 6 months of age, and they have totally non-developed limbs and other organ malformations, something like that. And Jackson Lab out there in Bar Harbor, it has mice models. That's what it's specialized for, that researchers can work on and study human disease. Because with mice, our genome even more similar. All right, so what we're going to look at now is very important to figure out how this gets converted into this. And don't be afraid. So
what was gene expression?
What
was gene expression?
Gene expression is like be all you can be. So here's a code, but there's different steps of actually making a functional protein. So gene expression is all the steps from DNA to protein. All these actions that the cell gets involved in order to make proteins. Because DNA is just like constitution, should be like that, and like that. What happens in reality may be like, slightly different. But the cell has to make it. Anyway. Now, here is the cell, and here is the nucleus. That's where DNA is kept, and it never leaves the nucleus because DNA is very, very precious. It's all the country records. No. So, somewhere else in the galaxy, far, far away, outside of the nucleus, there are little factories that are called ribosomes. That make protein. So I'm writing here, ribosome is a protein making factory. So, in order to make a protein, we need building material for the protein, right? What is the building material for the protein, once again? Amino acids. Different kind of amino acids are floating in here and need to be delivered to the factory in order to build protein. So there is also an enzyme, an actual routine manufacturer worker that puts those amino acids together, right here. The only thing that this enzyme, this factory worker doesn't know is the order. How should I connect those amino acids together, in which order, So we can make a working protein. And see the problem here, DNA cannot leave nucleus, it's kept here. But the instructions are needed here. So we have to have some kind of messenger that is bringing instructions from the nucleus to the ribosome. And we have such thing, mRNA. And I would like you to color this letter M with your coloring pencil and put messenger. Here. Because this is a message that is coming from the nucleus to the ribosome. You know, in former times there were like secretary running with paper between the offices. This is exactly this is a messenger that takes instruction from DNA and brings it to ribosome. Now, picture right here shows you that in the nucleus, RNA is synthesized just the way that DNA doubles. But in case when you need to reproduce, more DNA is built on DNA. In case when you need to synthesize, let's say, more muscle proteins, you will be synthesizing some particular protein. You need to get just that specific instruction for this protein, and you see RNA is being built right here. You see mRNA, messenger RNA transcript. You know what transcripts are, right? College transcripts, everything written somewhere. Now, why do we need RNA? See, DNA, RNA sounds like the same, right? Difference is one letter, reflects the differences in chemical structure, because RNA can leave nucleus, DNA cannot. RNA is one-stranded, DNA is double-stranded, that's why it cannot leave nucleus. RNA, one-stranded, and what guarantees it's being one-stranded is just, it's not T any longer. It's you see the letter U. This T base, remember Gattaca? So T is no longer a T. It gets replaced by uridine or another slightly different base. Just remember that it's slightly different. That's why you see now U instead of T. See, it's A and it's U right here. And it allows mRNA get to ribosome and deliver instructions from DNA. Now, so you look at this picture at home. Let's see. What is the difference between DNA and RNA? RNA being one-stranded and having U instead of T, which guarantees it's being one-stranded. Now, which RNA delivers a message from DNA to the protein-making factory, which is ribosome? That's easy. It's right in the name. Messenger. That's why we have little letter M in front of RNA, which shows its function.
What was the third difference? The third thing about RNA and DNA?
the
I have-
surname
I have-
differences.
I have can leave nucleus, single-stranded, what's the
Okay,
other one?
single, yeah,
The bases are different.
The bases are different, okay.
Means,
Oh, yeah,
okay.
I got it.
Good. So, oh my God, there's, I'm looking at my lecture note and I'm seeing a mistake. Okay. Now we need, we have ribosome, we have instructions. We need somehow catch out, you know, those amino acids that like fishes. Just keep swimming around ribosome, but we need actually to catch them and bring it to the ribosome. And we have another type of RNA, and I want you to write it in different color. And it's called tRNA, and T stands for transport. Okay, right here. Transport. So what it does, it brings amino acids to the ribosome. Because you know amino acids are just small stupid chemicals, they don't know where to go. So we need something that sticks to them and delivers them to the ribosome. And I'm putting tRNA here. And what I want you to remember about it is just transport. Transporting amino acids to the ribosome. So the question is, which RNA delivers the message from DNA to protein? We can factor it, mRNA. Which RNA? Because there are different kinds of RNA in the cell. So this is T, and I'm putting transport RNA. And there's another third kind of RNA, which is, you know, it's just, it's called rRNA, and rRNA is just a ribosome, because all the third kind of RNA actually makes the protein making factory or ribosome. That's why R in, you know, RNA, R stands for ribosome. It's ribosomic RNA. Now hopefully you won't get lost between three kinds of RNAs that ensure that DNA instructions will get converted into the protein. Now let's skip another question. I'm going to show you a short movie. And the next question is, what is translation? So far we have transcription, this process of making RNA on the base of DNA is called transcription. The process of making proteins, right here, putting amino acids together and actually getting the working protein is called translation. See the difference between the words transcription and translation. Transcription means rewriting information in the same language, nucleic acid to nucleic acid. Translation, you already kind of hear that it must be like interpreting to foreign language. So on ribosomes, the language of nucleic acid becomes the language of the protein. Okay? And that's what translation means. get this done.
Respond to the increasing amount of sugar in her blood by releasing insulin, a small protein.
Okay. I'm going to stop the movie and explain some parts about protein synthesis. But this little video clip emphasizes what happens in diabetes, or let's say you just gobbled up a big piece of cake and you have this nice feeling of eating a lot of sugar. This sugar, of course, will get into your blood, and we need insulin, which is a protein that will fish out all this glucose from your blood and puts it into your liver for storage. So all of a sudden, we need huge supply of insulin. Where does insulin come from? Hopefully not from, syringe. It comes from your body. Insulin is a protein. It needs to be quickly synthesized according to instructions that are in your DNA. And let's see how it happens. By the way, this one on the picture is a ribosome. The red thing, we'll see how everything works, is mRNA messenger, and the pink thing is a protein that each pink blob is amino acid. You see transport RNA that just deliver everything to the ribosome, and yeah, it sounds confusing.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin.
Okay, those pink blobs flying through the blood is the protein insulin. You see that it's composed of little pink blobs. They are amino acids.
In her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured.
Nucleus, look, we're moving toward nucleus.
The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription,
Okay, this is DNA. It's actually two strands there, but DNA is a long, long thread. So what you're saying at that point, DNA is being wrapped around special safekeeping devices. This is that scary thing, the yellow thing, is a protein that is going to transcribe DNA to RNA and make us a lot of insulin messenger RNA.
an enzyme zips along the DNA, forming RNA, shown here in red.
Look at that beauty. See, this is double helix of DNA, and what enzyme does, it pulls it apart because it needs to get to the code. And here's one strand of DNA, here is another strand of DNA. The red thing is mRNA being built from the floating RNA nucleotides that are being built according complementary to DNA strands.
RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA.
Okay, see how it's being done?
As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning.
don't concentrate on it. It's just a safety check that RNA is synthesized correctly and the instructions are precise. Okay, that's we're not studying this in detail in our class. Making precision work.
As a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand,
Okay,
forming a
that's for
tail.
stability.
The completed messenger RNA,
And look how it goes
mRNA,
out.
now leaves the nucleus.
So you remember that transcription is DNA to RNA, and RNA floats to the ribosome.
Message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying.
Look at that. The green blob is transfer RNA. This is amino acid attached to each particular transport RNA. Because you know, they are so highly specific, so one kind of transport RNA, like one kind of track, can carry only one specific amino acid.
The specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon.
Okay, this is important. This how, once again, the exact instructions, the precision of transferring instructions, DNA instructions into protein, I'm sure. As I said, tRNA is highly specific, and it has, you know, like a license plate on the end. So, and this license plate is called anticodon. So write it in your vocabulary, because that was a question that we missed. 3 unpaired bases in tRNA that match mRNA. Let's say mRNA here, and we have 3 bases. Remember nucleotide right here. Once again, it's the principle of being complementary to something. And that what ensures that what is coded in DNA is built into protein in exact order how it's coded. So this is a conversion of the code. If in DNA we call it a code, triplet code. So then mRNA is being built on DNA. Come on guys, you're going to be such a great specialist in molecular genetics, you believe. Because that's all it is. If you understand that, you can go into genetics, into like any neuroscience and so on. So this is called now the same 3 nucleotides. Let's say, once again, GATACA, GAT, that are called codon. And if you're talking about codon, it's automatically clear that you're talking about mRNA. So G becomes C. It should match T, but it's U now. 1, 2, 3. And T becomes A. That's what you see in mRNA. Okay. And then in tRNA, This is a license plate. Remember that there's amino acid, highly specific amino acid on the other end of tRNA. That is called anticodon. If you are talking about anticodon, you're talking about tRNA. And now C becomes what? Come on. Which letter should I put in here? G. Good. U becomes A. And A becomes not T. We're dealing with RNA. So it's like U. Okay? That's the only thing you have to remember. Well, the most important thing, the difference between RNA and DNA, which allows RNA to be single-stranded. That's what you see here. And see how similar they are? So this, and this is tRNA. Well, why don't I draw a track here?